All right, good morning, everyone. Happy Friday. How are you? Yeah. Tired? Yeah, me too. Yeah, we'll get through this, and then hopefully you guys are done for the day and can go home. If not, I'm sorry. I didn't mean to tease you, but I'm going home for the day. All right, so I know I haven't actually been up here. Well, I introduced myself at the beginning, but it's been a while. So if you've forgotten who I am, I'm Michelle Steros. I'm the course coordinator and a co-instructor. And as Dr. Michael mentioned, I'll just be doing the review today on immune. And then Monday I'll be doing digestion. He'll be back on Wednesday to finish digestive metabolism and then move on to urinary system. Okay. All right, so last class I set it up so that we actually went through the poll everywhere questions first, so just to make sure that we had a chance to review those. And that actually gives us a review of a lot of immune system already, but I still had time at the end to go over those review slides, and I expect it'll be the same here. Come back. Where'd you go? There we are. Getting oh, me and I broke it. All right, let me just try restarting. All right, sorry about that. I will warn you too, this is not my PolyV account, which is good for you. It means you can just use what you've always used, but it also means I'm not as comfortable with how it's set up, so this is an adventure. We'll get through it. But the question here is, during the activation of a T helper cell, the B7-CD28 interaction occurs to bring about antigen recognition, co-stimulation, proliferation, clonal expression. I think 26 is probably about most of you. Such a small group. All right, we've got the majority saying antigen recognition and a little bit of everything else. It's co-stimulation. Okay, so let's look at that. Here on this slide, we can see that your process of actually activating your lymphocytes is multi-step. So we have to have this antigen that's presenting, a T cell that recognizes it, so that's the whole MHC and CD story, depending on if it's an intracellular pathogen or if it's a presenting cell. But then in order to actually stimulate the clonal selection and being able to make more of these white blood cells that you need that have recognized a pathogen in your system, you need to have this co-stimulation by the B7 and CD28. Where's my molecules here? Okay, so those again, they're just transmembrane proteins. And it's just a secondary signal that's necessary for full activation. So we're not going to get into details of it, but I would recognize B7 and CD28. And just remember, this is kind of just a memorization one, but B7 and CD28, that's going to be your co-stimulation. B7, when that's on a cell, okay, that's not always there. That's one of those that only gets expressed in certain circumstances. So a good thing to remember about B7 is it's a signal that that cell has seen danger, right? So this has already had an activation event to put this B7 out there. Once that B7's there, then the CD28 can bind and you can fully activate. Right, so questions on B7 CD28? Good. So what from the T cell is actually going to help bring a cell presenting an antigen on an MHC1 closer to it? What is this T cell using as like a grappling hook to bring it in? Yeah. That's pretty good. Okay, CD4, CD8, B7, B8. Just kind of all over the place a little bit. Most of you said CD4. That makes me sad. That's wrong. Yeah. It's the rule of 8s. I wish I had a dry marker. But when you're trying to remember which MHC and which CD molecules are working with each other, follow the rule of 8s. MHC1 always works with CD8 because 8 times 1 is 8. Look at you, so prepared. Thank you. All right, so our rule of 8s. MHC1 will be recognized by a CD8 because 1 times 8 equals 8. So a CD8 will not recognize an MHC2. It's very specific. And for an important reason. CD8 is a T killer. You don't want T killer cells going around and killing antigen presenting cells, which are the ones that are going to be using the MHC2s. So an MHC2 works with the CD4 because 2 times 4 equals 8. Okay. And then if you just remember that MHC1 is on every single nucleated cell. It's the primary one. MHC2 is very specific just to professional antigen cells. Then that should help you remember what your CD8s mean, because your MHC1 is going to be presenting internal infections. You can't save that, which means you need to kill them. So a CD8 has to be your T killer. That means your CD4 is your helper cell. I'm going to give this back before I forget. I'll steal it. Thanks. Okay. So here's the slide where that was discussed a little bit. And you can actually see an example here of a CD8 protein that's coming in, recognizing an MHC1, pulling that cell in closer so that it can actually use its T cell receptor to check out the antigen fragment on the MHC. So questions on that? Okay, which one of these is not an antigen presenting cell? So which one of these would not be expected to have an MHC2? Macrophage, dendritic cell, naive B lymphocyte, mast cell. Okay. Again, all over the place. So macrophages, those primarily work to eat things, right? It's a macrophage, big eater. but once it eats it, it's going to break it up and it presents it. So macrophages are definitely antigen-presenting cells. They would have an MHC2 receptor along with their MHC1. Okay, so they are considered to be the link between the innate system and the adaptive system, because they come out in the innate system, they clean up the mess that was made, and then they present the first pieces that start the adaptive response. Dendritic cells, we didn't really talk about much. That one's just, you kind of have to know that it is an antigen-presenting cell. Naive B lymphocytes are definitely antigen-presenting cells. We told the whole story about how a naive B lymphocyte would present to a T helper cell. And then mast cells. Mast cells are static cells. Okay, these are not cells that will move through the blood or lymph. They sit in alveolar tissue, they sit in epidermal tissue, and they will react to an infection there, but they don't present because they can't go anywhere to present it, so it's not very useful. Okay, so this was a slide where he showed both dendritic and macrophages and the added naive B cells as being these cells that will grab hold of some kind of pathogen and then go around and showing off to everybody, look at what I got. Isn't this cool? Let's kill it. So that's an antigen presenting cell. MHC2, sorry, yes, MHC2 for an antigen presenting cell will work with a CD4 because you don't want to kill the cell that's doing a good job and just found this thing. You want to help it create more of itself because it's doing so good. You want it to go find all the rest of the pathogens in your system. Mast cells, on the other hand, those are going to be the ones that are releasing histamine in like your epidermis. Yes. So what's causing the mast cell to release histamines? They have receptors on their surface that will bind allergens or pathogens of different sorts, and it causes a whole cascade that ends up with release of that histamine. Okay, so it can also react to cytokines. It can bind those as well. So it recognizes PAMPs, yes, and then it releases DAMPs, essentially. You could categorize histamine and cytokines as DAMPs, reactions to that infection. Yeah, great question. Yes?
Do antigen-presenting cells ever lose their MHC?
Yes. Do antigen-presenting cells ever use their MHC1? Unfortunately, yes. There are certain infections like HIV that will target your immune system. They will get inside your immune cells, and then you're going to end up going through that whole process of breaking down their proteins and putting them on an MHC1, and then your other immune cells will be attacking those immune cells. That's one of the big problems in AIDS, but it's not the only virus that can do that. So yes, our white cells will use the MHC1 as well. That's not a good sign when they have to. Okay, when they are sneaky enough to actually attack our defenders, we're in trouble. Okay, so parasites are secreting what to trigger the generation of red blood cells? Is that going to be our renin story, erythropoietin, aldosterone, or angiotensin 2? Parasite. Yeah, maybe I should pronounce it like parasite, so it's not so confusing. Harry. It's easier to read, for sure. Okay. Yes, most of you said erythropoietin. Fantastic job. It is EPO that is going to trigger the generation of additional red blood cells. When we talk about renin, aldosterone, angiotensin, those are part of the RAAS system, RAS system, that is going to be a story from the kidneys in response to low blood volume. All right, so let's look at pictures. Okay, so here we have, oh hell, I can't see. Erythropoiesis. So you've got parasites being shown down here, parasites. that are reacting to low oxygen concentrations in the blood. So if it if it recognizes that there's not enough oxygen, your response to that is to create more red blood cells so that they can go pick up more oxygen from the lungs. So you release that EPO into the blood, it travels through the bloodstream to the red bone marrow, and it will bind to a proerythrocyte. That proerythrocyte will then be able to mature into a normoblast. And a normoblast is anything but normal because it will kick out its nucleus. Right? It's one of the few cells in the body that does that. Once it kicks out the nucleus, everything else starts falling apart around it. Okay, the nucleus sort of holds everything together. So this normoblast is going to continue to develop and everything else will break apart. So you get to a point where if you actually stain this, all you would see left of the ER is just tiny little scraps here and there. Okay, so they named it a reticulocyte, meaning it just has a few pieces of reticulum in there. That cell then moves out of the red bone marrow into the blood, and it matures into an erythrocyte, a full-grown erythrocyte that carries all of the oxygen to all of your tissues. Okay, so questions on pericytes and EPO. Yes?
Wait, at what point is the nucleus actually discarded?
Which one, at what point is the nucleus?
Discarded from the red blood cell before.
So it's at the point of ejecting the nucleus that we would consider it a normoblast. Anything else? All right, so if you're in the red pulp of the spleen, what kind of cells are monitoring the oxidation state of your erythrocytes? Dendritic, basophil, macrophages, or lymphocytes? All right, we got everything. And we're still changing. All right, let's walk through this. So normally in your capillaries, you have gaps, right? Capillaries are a single layer thick so that you could diffuse things across, and you've got these gaps in there. You could push a lot of your plasma out. You could push small molecules out. You cannot push out cells in most areas of the body. There's one exception though, and that is in the spleen. In the spleen, we have fenestrated capillaries that have gaps big enough that you can actually push those red blood cells out so that they can go through a check. You could consider it like a security check. You have to make sure they're still able to function and do their jobs properly. So you push that red blood cell out into the red pulp, and there are macrophages there that will check the pattern on their surface. So all cells have glycosylation patterns on their outside, and that includes red blood cells. They existed before you got rid of all the machinery, so they're still there. But they can't fix them, they can't update them, because they don't have the machinery anymore. So they don't last a whole long time. And you can tell when they're getting old because the glycosylation patterns start getting oxidized. So the macrophages read that glycosylation pattern, determine whether they're still functional and they can go back into circulation, or whether they're too old and need to be retired. If they determine that they're too old, they will eat them and they will break all the parts down. They will recycle everything they can, amino acids, the sugars, the carbohydrates, and the glycosylation patterns. Most of the hemoglobin is junk, except for the iron. The heme itself will save that and reuse it. But all of that junk is going to be sent to the liver for processing. So that's your bilirubin, biliverdin story that you have to get rid of. That's the stuff that you don't keep. But it is macrophages in the red pulp. Everything else here, all of these other, not really basophil. Well, okay. Everything else here you would find in white pulp of your spleen. That's where you have germinal centers, where you're actually looking for pathogens and microbes. Yes? That's okay if you think of it. Raise your hand again. Anything else? Yes. I'm sorry, I didn't catch that. Lymphocytes are, you're asking if they're like naive cells? Lymphocytes are actually any of your B or T cells. So we really have kind of a broad category breakdown of white blood cells. Lymphocytes, which are your B and T cells in the adaptive system, and leukocytes, which are everything else, like your granulocytes, your macrophages, megakaryocytes, all that kind of stuff. Right? And then we can like sub-break it down from there. Did that answer your question? Okay.
When we break down red blood cells and we want to keep iron, do we save the whole heme group or just the part of the same iron?
Do we save the whole heme group or just the iron? I have no idea. No idea. I know we save the iron. I don't know if we save all the parts. It would make sense that we would just keep that heme and be able to make a new hemoglobin structure from it. But I would guess it actually gets broken down to its component parts and then put back together again later. That tends to be how our body works in most cases. Yeah. Okay. Oh, there we go. Here's more erythropoiesis story. So that EPO, again, it's going to attach to a proerythrocyte. That's going to activate it to become a normoblast. That's kicking out its nucleus. Your reticulocyte then moves out of the system and becomes an erythrocyte. Then you break it down in the spleen. The macrophage will eat it. Okay, I think we're good on that. All right, so a Plasmodium falciparum is a parasite that causes malaria. And the question is, which of the following cells are going to detect that Plasmodium? So is that going to be basophils, eosinophils, neutrophils, megakaryocytes? Bless you. So this is kind of an example of a question where we can give you something that you've never heard of before, because we're not actually expecting you to know anything about the Plasmodium falciparum. We just want you to know, do you recognize which of these cells usually detects a parasite? And let's see. Most of you said neutrophils. I'll give you a second chance. Yes. Okay, eosinophils shot up. Stay there. You're perfect. Eosinophils. Yes. Okay, neutrophils are going to be the ones that are going to be involved in the inflammation process. We talked about that as an example in the epidermis and the dermis. Those are going to be recognizing bacterial toxins. They don't really function much for recognizing parasites. Basophils, that's going to be allergens. And megakaryocytes actually break up into platelets to create blood clots. Okay, so eosinophils are going to detect Plasmodium. So here's the slide where we talked about all the different cells, lymphocytes and then our granulocytes. Don't memorize this entire slide, but recognize the difference between the cells. Which cells function as attacking parasites, which ones are going to be involved in inflammation. You could maybe look at like the differences in the nucleus. This one has 3 lobes, this one has 2 lobes. I don't know. I feel like that's a detail that's not terribly important to the story. I wouldn't ask about it. I can't swear that he won't. Sorry. All right. Okay. So eosinophils, they're going to attack large parasites by releasing compounds to try to kill them. It's pretty much what all these granulocytes do. They're releasing some kind of compound to try to kill whatever they're targeting. All right, so now which of these cells detects allergens? Lymphocytes, eosinophils, neutrophils, or monocytes? All right, let's talk about this. So, lymphocytes, again, are your B and T cells. They're going to respond to pathogens. They don't typically respond to allergens because although our body sure seems to act like it's something bad, allergens are not necessarily harmful. They're not pathogens. This could be something like pollen in the air, okay? It's not going to hurt you. Your body's just overreacting. Okay, so we don't like having allergies, but it's not actually a sign of like an infection. We would not incorporate lymphocytes into that story. Eosinophils, we said that was attacking parasites, right? So I could see where you'd be like, yeah, that doesn't make sense. Neutrophils are going to be the ones that go into the inflammation story and release toxins, or sorry, release, well, yeah, it is. It's free electrons to attack bacteria, not allergens. Then monocytes. Monocytes are just baby macrophages, right? Immature macrophages that haven't gotten to the tissue yet. Those eat things, usually after they're dead. Okay, so probably not a good idea. So you're like, well, what's the answer? We said that basophils were in charge of allergens, and we didn't give you that option. Because the correct answer here was actually eosinophils. So that's not its primary purpose, but it is what it tends to do. And one of the reasons for that is because in most first world countries now, our sanitation is good enough that we don't typically have a lot of parasitic infections. So eosinophils are bored. Okay, they're like a working dog that somebody adopted and tried to turn into a lap dog. It's not going to go well. They're going to have all this energy, nowhere to put it, and your house is going to be destroyed. That's essentially what an eosinophil is doing. It doesn't have a parasite to attack, so it's just going around trying to find something. It sees pollen, and it's like, okay, whatever, that'll do, and it attacks. Okay, so it's not meant to work on allergens, but it does. And in fact, this is the allergies that get really nasty for us, that overreaction that we don't need. All right, so questions on your different granulocytes. Know that basophil is going to be allergens, eosinophil primarily parasites, but it will also cause allergic reactions. And then you've got neutrophils and inflammation. Monocytes are your macrophages and antigen presenting cells once they turn into a macrophage. And then lymphocytes are your adaptive system. Okay, so I told this story in the last class, and you guys can look it up. I wish I had now. I would have brought it in. But I actually read a scientific study where this guy had infected himself with parasites intentionally to see if keeping the eosinophils busy and happy would actually reduce his allergies. I don't recommend this because parasites have a lot of other side effects. You know, they release things that will cause damage to your tissues. So I don't care how bad your allergies are, that's never the solution to go eat worms. Just take your Flonase instead and deal with it. Okay. So oh yes. Did it work? Yeah, it did. Not only did it reduce his allergies, he lost weight. It was a digestive worm. Yeah, so that's terrible. Please do not do this.
Or like if someone wasn't losing weight, to just like inject a parasite and then bye-bye baby belly, then you should not be.
They Did not. Really?
I think they did.
I guess it's no crazier than HGH and whatever else you want to inject.
Weird stuff in the past.
Theoretically, like if you decide to be like molecularly engineer a parasite that like wasn't harmful to your body, that you could actually like prevent allergies to some extent.
Could you genetically engineer a parasite to be relatively harmless, not to release anything, probably not to reproduce because in and of itself just taking up space can cause damage? Would that be I still think that anytime we're putting another organism into our body that we're not already well adapted to, like our bacterial biomes, I think that could probably lead to a horror story at some point. So I still think it's better just to try to block the allergic reaction at our own molecules rather than, plus, it's just kind of creepy to eat worms. I don't know. Okay, so what kind of surface molecule or marker is expressed on our blood cells? Cytokines, clusters of differentiation, or CDs, pattern-associated molecular patterns, or PAMPs, or the damage-associated molecular patterns, or DAMPs. Okay, most of you said pattern-associated molecular patterns. That'd be bad. That would be so bad. PAMPs are located on pathogens. So remember the P's go together, right? PAMPs on pathogens. PAMPs are a sign that something is not self. And that means your body's going to attack. So if you had these on your blood cells, your body would attack all your blood cells. It's definitely not PAMPs. Damage-associated molecular patterns, those are secreted. Those are reactions to an infection. Those would be things like cytokines and histamines. And cytokines, obviously, it's kind of damp. And then clusters of differentiation, or CD molecules, those are what differentiate each blood cell. Every blood cell has its own CD marker, a CD protein. And there's literally, there are more CD markers than there are blood clotting steps. Okay, you probably haven't learned clotting. It's a joy, let me tell you. But there are a lot of CDs. Okay, we're not going to make you learn all of them. We make you learn CD4, CD8, CD28. That's it. But know that all blood cells are going to have different CDs on them, and that helps distinguish which ones are which. Okay, so here is the slide where we actually went through the differences between those PAMPs, those stamps, the CD molecules, and the cytokines. So questions on those? Good. So what is it being expressed by a professional antigen presenting cells that has already seen danger? It's been activated before. So I'm going to be CD4, CD8, B7, B8. Good, most of you said B7. Fantastic. That's the one we talked about that is going to be the co-stimulator with CD28. And we only express B7 on antigen-presenting cells that have already seen danger. Okay, so that's part of the stimulation process. So this was this slide where we can see our B7 and CD28 interacting as part of that co-stimulation. Sorry. All right, so B7 is essentially our been there, done that sign for a cell. Okay, here's another one, SARS-CoV-2. It's an RNA virus that causes COVID-19. I'm sure you guys have heard lots about it by now. If SARS-CoV-2 gained access to alveolar epithelium, you would expect the innate immune response to mount a response that secretes. So if you get infected with COVID, what is your innate immune response going to release? Histamines, interferons, interleukins, chemokines. So this is another example of a question where we're going to talk about something that we haven't really gone into detail in this class, but you don't need that information. Really, all you have to do is go through this question and pull out the key terms. Pull out the stuff you know we did talk about. So virus, you know what a virus is. We know alveolar epithelium. We know innate immune branch. So basically, you could boil this question down to, what does the innate immune system do in a viral infection? Most of you said interferons. Fantastic. Okay. There are two innate stories. There is the viral and the bacterial. Okay, so in a viral infection, it's an interferon story because the virus is actually going to get into a cell. Viruses cannot survive outside of a cell. They're obligate intracellular organisms. They need our machinery to be able to create new proteins and replicate. So when you recognize that this cell has a viral infection, the first step, before you're putting this on an MHC1 and getting the adaptive system involved, the very first step is your neighborhood watch program, where you're going to release these interferons from the cell, and it's going to go knocking on the doors of all the neighboring cells and say, hey, I'm infected. You need to make sure you've locked and closed all of your doors and windows. Don't let anything in. Stop making so many proteins in case it's already in there, so you can slow it down until the adaptive system can get here and actually attack and kill you. Right. Unfortunately, there is no way to save an infected cell because you can't put an immune cell into another cell, like you can't do cell in cell. So you just have to kill it. So what was that? I heard apoptosis. Natural killer cells and apoptosis? Yes. So one of the ways that you can actually kill an infected cell is with natural killer cells. They have a tendency to go after, I don't know, I always hear them in response to like cancer. Okay, but I think they also work with viral infected cells as well. So you can either go through the pathway. We only gave you like one example to keep it simple. Immunology is way more complex than we're leading on here. And there's a whole class, a fantastic class that you guys could take on that. But yeah, you can have this whole story we told you about perforins coming and poking holes in the system, or you can have something that actually signals for a slightly more organized apoptotic event. All right. So which region of an antibody would you expect to find embedded in the plasma membrane of a naive B cell? The constant region, the variable region, stem region, none of the above. Most of you said variable region. No, that's so sad. Okay, so let's look at an antibody. I think we gotta have a picture here. Here's an antibody. You've got a variable region on it, and then you've got a constant region. Okay, so this constant region here, and then these parts, the Y part, it contains the variable regions. Constant means that it's constant across different antibodies, right? So you can make different antibodies. You can have an antibody that attacks a SARS-CoV-2 virus. You can have one that attacks a cold virus, right? Any kind of infection you can imagine, bacterial, anything else, even parasites. You'll have specific antibodies to attack it. So it changes the portions here in the variable region to be able to match different pathogens. You would not ever want to stick that in your membrane because then it would be buried and it wouldn't be able to find the pathogen. Now these constant regions that are the same across, you don't need those. Those are really just the foundation, the concrete that you're going to build your variable regions on. So those are the ones that make the most sense to plug into your membrane and then wave the variable regions around to find the pathogens. Make sense? As for stem cell or stem region, I think he made that up. Never heard anything on an antibody referred to as a stem region, so. Okay, any questions on antibodies? Yeah. Sorry? The constant region is the one in the plasma membrane, yeah. Of course, that's only true in the naive B cells. Once you go through clonal selection and now you have plasma B cells, you're actually going to be secreting those antibodies out. They're not embedded into any membranes. All right, so which of these molecules are secreted proteins that function as signaling mechanisms via autocrine, endocrine, and paracrine signaling? Cytokines, CDs, PAMPs, or DAMPs? Most of you said cytokines. Good job. Cytokines is a really broad category. It encompasses everything from chemokines to interleukins to thousands of different molecules. Okay, but all of them have the same basic function. They're meant to signal. How they signal may vary. Like chemokines are chemical attractant versus cytokines might actually bind to things to create a signal, but they all have that signaling function. The CDs, remember, that's going to be the transmembrane portion of a cell, of that blood cell that will help differentiate all of your different blood cells. Your PAMPs, that's going to be on a pathogen, P for pathogen. Transmembrane mostly, or membrane associated, this could be like your LPS on bacteria, but it could also be bacterial toxins that are released. Okay, so it could be secreted. And then DAMP, of course, is your reaction to the infection. None of those are specifically signaling except for cytokine. So cytokine was correct. Nicely done. So our same slide that we've seen before. You should have this memorized by the time we're done. Oh, that's it. Okay. So do you guys have specific questions, or do you want to kind of spend the next few minutes that we have doing a review of the overall slides? Go ahead. So the cluster differentiation is a transmembrane protein that exists on all blood cells. There are, I mean, they keep naming them. There's so many of them. We only ask that you know the CD4s, which are going to be on your T helper cells, the CD8s, which tell you that it's a T killer cell, and the CD28s that are part of the co-stimulatory process. Okay. Anything else? All right, let's go all the way back to the beginning then. We can skip through this pretty readily since we've covered a lot of it in the review. But in terms of components of the blood, again, I'm not worried about percentages. I want you to recognize what is in each portion of the blood and recognize why we can actually get these to separate this way. So when you spin a whole blood sample very quickly, it's going to separate by density. Heavier things will fall to the bottom and the lightest things on top. So you're going to see this portion down here that we call hematocrit, and it's full of red blood cells. That's because these all have these heme in there, okay? All this iron is heavy, and it will make this the heaviest portion to drop on the bottom. Any other cell that doesn't have heme in it, so all of your white blood cells, they're all going to be in this very skinny little buffy coat portion. Then when you get into plasma, that's your water portion. That's ions, that's microbes if you've got any. That's going to be secreted antibodies. All are going to be in there because they're so much smaller and lighter than a cell. They're going to be that top portion. Okay. Again, percentage doesn't matter here. It's really important for clinical applications. This is a good way to be able to tell if you have an infection, if you suddenly have a much higher percentage of your white blood cells, if you've got a problem with your erythrocyte generation or breakdown, they have to be in very tight ranges. So if you look at this and you see that it's outside of those ranges, it gives you an idea that there's an issue that needs to be addressed. Yeah?
So formed elements is the buffy coat and the erythrocytes.
Formed elements, buffy coat and erythrocytes. So you're going to have formed elements in both, yes. Erythrocytes are going to separate out into the hematocrit, and then all of your other white cells, including platelets and other formed elements, would actually be in the buffy coat.
Wait, I'm a little confused.
Mhm.
Is the big category the formed elements, and then it divides up? Or,
How did he define formed elements? Does anyone know?
I don't think it is.
He didn't? Then I wouldn't particularly worry about it. I usually consider formed elements to be the portions, the parts of the immune system that don't have nuclei, like your platelets and your red blood cells, but I've actually seen a definition that included all white blood cells, including nucleated, so I'm not clear on it myself. I certainly wouldn't ask you on it. Okay. Okay, structures. Again, know their primary function. You could probably, wouldn't hurt you to know the lobes. 3 for your neutrophils, right? 2 for eosinophils. Know that your red blood cells are usually concave. I don't care that you know the structure so much as the purpose of that. Okay, so that biconcave structure is going to allow it more surface area to be able to do that oxygen and carbon dioxide exchange. And it's also going to allow for this to be able to move more readily through capillaries. Okay, so remember that it's always form for function. Okay, we talked about this. Did you guys have any questions about EPO to breakdown in the red spleen, or red pulp, sorry, of the spleen? Good. Okay, hemoglobin structure. We're not going to get into the nitty-gritty details of this. You'll get that pleasure in biochemistry if you take it. You guys should just know that you have 4 subunits or 4 chains. You can call them either. In adults, it would be 2 alpha, 2 beta. Each one of those is going to have its own porphyrin ring with a heme that contains your iron. And it is the iron that the oxygen will bind to. So every hemoglobin can bind 4 oxygens, and you have hundreds of thousands, if not millions of them per erythrocyte. Okay, so we can carry a lot of oxygen. Differences in stimulation of your white blood cells. This is going to be colony stimulating factors. Okay, I don't think you really got into detail about the differences between them, but it's fairly simple. If you do see a CSF listed, the letter in front of it kind of tells you. If it's a G, it's granulocytes. If it's an M, it's a monocyte macrophage story. And then we just have multi, which pretty much works on everything. Okay, EPO would actually fall under a multi-CSF. So that is your stimulating factor for your development of your blood cells. Okay, doping, I would know these 2 stories. They're very similar. One is just injecting EPO into someone to increase their red blood cells and increase their oxygen carrying capacity. The other one is packing or doping, where you actually create all of the red blood cells outside of the body and then just pack them in. Okay, so you're just skipping the step of the EPO. Right. Neither one of them is a good idea. There's not a lot of evidence that it's all that helpful in endurance, and it can cause a lot of problems. It can lead to thickening of your blood, which can lead to things like blockages, clots, strokes, things like that. So not worth it. Don't do it. All right. We'll leave it at that. Have a great weekend, and I'll see you guys on Monday.